Hi everybody, I'm Nuria Sanchez from the University Complutense of Madrid, and I'm going to talk about canories. Yeah. Okay. All of you will know what is the Meiofauna. It's comprised the animals with a small size that lie between the sediment particles and inhabit the first centimeters of sediment with a patchy distribution. Most of the currently recognized animal phyla has Meiofauna representative, but only 6 of them are exclusively Meiofauna groups. Canorinx is one of these exclusive Meiofauna phyla that moreover only appear in marine sediments. Its name comes from the Greek kino, move, and rinkos, smooth, referring to the typical movement of the animal when it's alive. The first canorinx was discovered by the naturalist French Dujardin during the 19th century, and currently the field comprises 210 species, divided in 2 main groups, Cyclorhagida and Alomalorhagida. All of them have a small size and share a similar external morphology, divided in 3 major regions: head, neck, and trunk. So, sorry, here. It's an elongate body divided in these 3 regions: head, neck, and the trunk. We are going to take a look to each of these regions because all of them has taxonomical relevance for the group. The head is divided in the mouth cone, where the mouth is located, and the retrograde. All of them with cuticular appendages radially arranged. The cuticular appendages of the mouth cone are named oral styles, and their shape has a taxonomical relevance for the group because they may appear as articulated pieces or may appear as a single piece, or even they may be fused in some groups. The cuticular appendages of the introvert are named scalids, and they appear arrangement in up to 7 concentric circles around the mouth cone. A special kind of scalids are named trichoscalids and always appear in the last ring, and they may be directly attached to the introvert or be attached by partial plates with taxonomical relevance because these plates only appear in one order. The neck consists of a ring of plates named placids that form the closing system when the introvert and the mouth cone is fully retracted inside the trunk of the animal. And their distribution and development also has a taxonomical rule for the group. And the closing system, depending on the development and the arrangement and number of these particular plates, make that the system close radially or bilaterally. The trunk consists of 11 segments in adults because the juvenile stage may present a few number of segments, and the trunk is covered by cuticle. And the arrangement of these segments, as well as the profile of the trunk in a cross-section, has also taxonomical relevance for the group. Actually, the phylum is closely related with Priapulids and Lorififera, forming the Scalidoptera group. The current classification has been recently updated after the phylogenetic works made by Sorensen and co-workers. According with the result, we have 2 main clades that mostly agree with the traditional classification for the group that divide canorinx into Cyclorhagida and Omalorhagida. And I say mostly agree because Phascistideres and Dracodelidae, 2 families, appear more closely related with the remaining Homalorhagida than with Cyclorhagida. And Dracoderidae was traditionally considered as Cyclorhagida family because they present some morphological characters considered as apomorphic for Cyclorhagida. According with the result, it seems to be more closely related with this other group. So therefore, the classification was made, and now the phylum is divided into two classes, Anomalorhagida and Cyclorhagida, being these two families, Franciscideridae and Dracoderidae, including into the Anomalorhagida. So now we have two classes and 3 orders, 11 families, 24 genera. Okay, now we know how a canary is and how they are classified. But how we can classify a specific specimen when we find a canary in our sample? So we have 3 main morphological characters useful for taxonomy: the trunk shape, the segment arrangement, and the cuticular character of the trunk. Let's see the first one. The trunk shape is useful to discriminate between the 2 classes of the group, because Cyclorhagida has a slender shape and a round or hard shape profile in a cross section of the trunk, whereas Anomalorhagida has a rectangular outline with a triangular cross section. The profile in a cross section of the trunk is of major interest for the group because the cuticular characters that we are going to use for taxonomy, their position depends on these shapes. As we can see, the position where we can find these structures are the same in the dorsal side and in the ventral side of the 2 groups, but we can find some difference in the lateral areas. So we have 2 standardized systems of positions depending of the profile of the animal, one for Anomalorhagida and one for Cyclorhagida. The second point, the segment arrangement. So it's going to be useful to discriminate until the genus level. The segment may be arranged as a closed ring or be divided into plates, being the dorsal one named the tergal plate and the ventral one the sternal plate. And this ventral plate may be undivided or may be divided into 2 plates. The arrangement of the segment 1 and 2 are those that more vary among canary. So we can find a segment 1 as a closed ring in all Cyclorhagida species, whereas most Anomalorhagida species have a segment 1 divided into plates, and this ventral plate may be divided with intermediate stage. And the segment 1 always appears as a closed ring in Cyclorhagida, whereas some groups of Cyclorhagida may present an intermediate stage until reach this other arrangement. The segment 3 to segment 10 are usually arranged with this shape in 1 tergal and 2 external plates, but we have some exceptions, and same for segment 11. And how about the cuticular characters of the trunk? So there are some characters that we can use to discriminate among the highest taxonomical level, because, for instance, Cyclorhagida always show spines in the mid-dorsal line in much of the trunk segment, whereas Anomalorhagida usually show other kind of cuticular structure different than these spines. And also the tubes are useful because Cyclorhagida usually show this kind of tube, but Anomalorhagida only show tubes in segment 1 in males. And the character that they present are zeta instead of this tube. Moreover, there are other cuticular characters that we can find along the trunk, and their distribution, kind, and number are absolutely needed to identify the specific specimen at the species level. So we have a lot of morphological character useful for identifying specimen. Well, now we are going to take a look to each of these two cases. The Anomalorhagida has 4 families: Neocentropidae, Pycnophidae, Dracoderidae, and Franciscideridae. So, as we previously said, we can discriminate between the groups by the term shape, being triangular in Neocentropidae, Pycnophidae, and Dracoderidae, whereas Franciscideridae has a round or heart-shaped profile. And what about the segment arranged? Neocentropidae and Pycnophidae has the segment 1 divided in 1 tergal and 1 external plate, whereas Dracoderidae and Franciscideridae has a close ring arrangement of the first segment. And for the remaining segment of the trunk, they appear divided in 1 tergal and 2 external plates in Neocentropidae, Pycnophidae, and Dracoderidae. But Franciscideridae has all the segments arranged as a closed ring. And we have another exception in Neocentropidae because the shape of the 11th segment is like divided in 1 tergal and a single ventral plate. We have more characters that are useful to discriminate at least between Neocentropidae, Pycnophidae, and Dracoderidae and Franciscideridae. And this character is the presence of a spine in the mid-dorsal line that appear in all trunk segments in Dracoderidae and Franciscideridae, but they are restricted to the last trunk segment in Neocentropidae and absent in Pycnophidae. So, and then we have 3 genera in Neocentropidae, 2 in Pycnophidae, but this is going to change in a couple of months because we made a phylogenetic analysis of the family, and now it's going to be arranged in 7 genera. So, that's all for the class Anomalorhagida, and now we are going to take a look to the Cyclorhagida with 3 orders: Xenosomata, Kentrorhagata, and Echinorhagata. What about the trunk shape? All of them with a trunk shape profile, like heart shape. And what about the segments? Well, segment 1, as we previously said, always arranged as a closed ring, and we start to find difference as for the segment 2, that it's always divided into plates in Xenosomata and Kentrorhagata, whereas some Echinorhagata have the arrangement of a closed ring, or maybe divided into plates, and we can find also some species with intermediate stages between the closed ring and the totally divided. And we have the order Xenosomata, we have 1 family, Kentrorhagata with 5 families, and Echinorhagata with a single family. And here we can find some diagnostic character for each of the orders, like the presence of this mid-terminal spine referred to this spine in the last trunk segment that appear in Xenosomata and also in Kentrorhagata, but it's absent, it should be here, it's absent in Echinorhagata. We have other morphological characters that are useful to identify the order. So this is the order Xenosomata with 1 family and 1 genus with the diagnostic characters, and this is a drawing of one of these 2 species. And Kentrorhagata, as we previously said, with 5 families. We have a lot of different morphotypes in this order, but as I said previously, we can find some difference in the segment 1 and in the presence of the mid-terminal spine related with this The lateral-terminal spine is in length. And lastly, the order Echinodermata with 5 families. And this was the order that the arrangement of segment 2 is useful for the taxonomy. And that's all about the taxonomy of the group. And now we are going to talk a little bit about the distribution. Well, chinorrhins appear from the polar regions to the Ecuador all around the world. And as you can see here, there are some places that are very well sampled, and this is because this distribution is more than a reflection of the samples that are real distribution of the animal. As you can see here, you have really well sampled area, as well as in the Italian peninsula and in Korean peninsula and in the west coast of the USA. And for the deep samples, we have only data about these localities, because the green dots refer that the chinorrhins were identified and the data were published, but the red colors refer that we have chinorrhins in this area, but the data are unpublished yet. So all of these points, only this one refers to deep samples. And in these deep samples, we found these species. So, Pycnophis avisorum is the deepest chinorrhins found that was identified until to a species level, followed by these other 3 species, Mystophias abyssales, Pycnophis nubilis, and Pycnophis farinelli, collected in the Guinea basin. Are these species? All of them, these 2 belongs to the Pycnophis genus, but show some strange character. And this is the other species collected in the Guinea basin with weird character. And in fact, this genus was the first genus of Anomalorrhea, described from more than 13 years ago. So it was very important, this discovery in the deep sea. So as we can see in the graphic, the number of described chinorrhins has been increasing during the last 10 years, but the diversity of the group is still poorly known. We know the 20% of the chinorrhin diversity, and in the other slide we saw that only in a very few samples collected for the deep sea we had 16 new species belonging to 3 new genera. So that's what means that we have a very high diversity of chinorrhins in the deep sea, but it's still mostly unexplored. We have to make a big effort to describe all these species, because what we expect is that we have not only new species with character, but we have maybe new genera or even families, as the Mystophias case. And what about ecology? Well, we have very few data about the ecology of chinorrhins, because we are a really small community of research interested in the field. We are right now maybe 10 or even less research working in chinorrhins. So we have a lot of work, and maybe that's the problem, that to justify that the few papers that include some information about ecology of chinorrhins only include that they have chinorrhins speak until the phylum level. They even described not to a species level, but just at least to the class level, but no, we are very few people working in this, and maybe one of the problem. But we know something about the ecology. We have some data about depth. We know that chinorrhins may appear since the intertidal zone to the deep sea. And also we know that we can find chinorrhins in several kinds of sediments, from mud to the coarse sand. And we carried out a study looking for some relationships between depth and sediment with chinorrhins, and we found that some species appear in all kinds of sediments, but at the same time there are some other that seems to be related with the kind of sediments. And also the same for the depth. We have some species that appear in a wide range of depth, but other ones seems to be related with shallow waters. What about the abundance? It's usually that chinorrhins are between 1 and 8% of the total Meiofauna, but sometimes they are quite abundant after nematodes and copepods. And for the salinity, they can have it into a range between 3.68 and 44% of salinity. And also they can have it in waters below 0. And also we have chinorrhins in hypoxic conditions. And what about the feeding? We know that they feed on detritus, bacteria, and alga and diatoms. And their abundance seems to be in phase with the density of diatoms and bacteria. And also we know that some polychaetes, bivalves, and crustacea food chinorrhins because some research found some specimen inside the gut of some of these animals. But for instance, we don't have any data about light, pH, how the contaminants affect to the abundance of chinorrhins, or even if we can use chinorrhins as biomarkers or bioindicators. So that's all. Thanks everybody, thanks Gordon and the organizing committee, and Martin Sorensen from the University of Copenhagen and Fernando Pardos to sharing some of the pictures of this presentation.
Any questions?
Just curious, you're talking about the distribution related to the salinity, right? You say it's only up to 44 per thousand. That's the up maximum.
Yeah.
So you have never found them in the hydrothermal vent or cold ship environment?
That was the highest data for salinity. It's appeared between these range, but the highest salinity is the 44%.
So any people look at the sample from a vent or the ship's environment, where is the salinity way much higher than this?
I don't know. So
I suspect not. Because I don't think Meiofauna is a big in this area, so I suspect that.
I suspect they will possibly will be there.
Yeah,
You know?
I agree.
yeah.
Other questions? Pedro?
So from what we know for the moment, can you say something about the distribution of the diversity with depth?
We don't know. The problem here is that we are in the very first step of everything related with chinorrhins. So we have, for instance, the specimen that I saw for the deep sea. The 3 species were collected in the same point. So 3 species in a core for chinorrhins, it's a high diversity. So But I think that we could expect to find several species in a multi-core sample because we carry out an extensive subcarpine forest in the Iberian Peninsula, around the Iberian Peninsula, and we have 51 species around the Iberian Peninsula. So imagine how can increase the number if we know the diversity of the deep sea, but we don't know anything. So I can't say anything else. As we say, we are in a very first step. So.
Can I ask a question? Is it possible that the reason we don't know is because people don't recognize kind of rings and samples?
Yeah, that's the reason why I put the video.
Yes.
Because they have a really
Pay attention,
easy
it's
to
important.
recognize the movement. It's really easy to recognize because when you know how they move, it's
But what about dead ones? Because often the sample sets come in.
yeah. It's more difficult. I think that it's always happened with all the groups.
Yeah, sure.
But
If you take a multi-core, is there things that we should do to make sure that we're looking for kind of rings? Because we're under-sampling them because we're just not seeing them, or?
yeah, so I think that it's easy to check if they have spines, but sometimes they can be confused with copepods. When somebody is starting with a group, some students say, I have a canary. Yeah, no, it's a copepod. Move the animal because they have the ventral side. So
Yeah.
Just a comment, possibly need to check with Greg Rose. They published a new paper recently. Number of the small phylum being, I cannot say this month, it's basically no longer exist. They all been merged into a group, the crustacean. It's a big group. They call group. They no longer call them a phylum. They no longer call them an order either.
yeah.
Lots of this is the new taxonomy happened right now. So you maybe check this again, whether this is the phylum being considered or not.
Yeah, but for instance, with the phylogeny of the family Pycnophidae that I say, it's like it's a mesh because they put everything inside. But now we are starting to take a look inside this black box, and we are starting to arrange everything. So maybe we are going to increase the number of families in general in a couple of years. So.
Any other questions? We'll move. Well, thank you very much, Nuria.
Thank you.
Very interesting.
Thank you.